Chapter 24, Immunity and Host Defense. The objective is students demonstrate the fundamentals of clinical microbiology, host-parasite relationships, and the fundamentals of immunology. We left off in Chapter 23, starting to talk about innate resistance to infection. So I'm going to include that here before we talk about the overview of immunology. Hosts have innate resistance to most pathogens. Excuse me. Host defenses can be split into groups. Specific host defenses that target an individual species, and non-specific defense mechanisms that target a wider range of invasive pathogens. We're going to talk a little bit about these non-specific defenses right now. Natural host resistance is conferred by many different factors. Some species are not infected by certain pathogens, many animal pathogens are not human pathogens, and cold-blooded pathogens rarely attack warm-blooded animals. First and foremost, the structural integrity of tissues is a barrier. Skin and mucosal tissues are barriers to infection. Intact surfaces usually prevent colonization. Damaged surfaces promote colonization. Skin also secretes acids at lower pH which inhibit growth. Secondly, cilia and mucosal tissues causes pathogens to be expectorated or swallowed. Thirdly, acids in the stomach act as a barrier to to invasive species trying to get into the body and grow. Fourthly, cells that make it past these barriers must complete with normal flora in the gut that we talked about earlier. And then they have to hang on because most things are swept away within 24 hours. In addition, tears and other secretions contain lysozyme, which will lyse bacterial cell walls. The blood contains beta-lysins, which bind and destroy microbial cells. So, pathogens are tissue-specific, meaning one avenue of infection doesn't fit all types of parasites. And when a host becomes compromised, it means that the host has lost one or more of the defense mechanisms I just mentioned. Hospital patients are often compromised hosts. Many hospital procedures are invasive and leave avenues open to infection and colonization. In addition, the stress of surgery diminishes resistance in patients. Compromised hosts exist outside the hospital as well. Smoking, no sleep, alcohol and drug use, and poor nutrition all contribute to lowering resistance to pathogenic bacteria and viruses. And then infection by other diseases such as HIV compromise the host as well. What makes us susceptible to disease? Well, age is an important factor. Infectious diseases are more common in the very young and the very old. The young do not have immune systems or natural flora to impede infection. The old no longer can mount as good as a counterattack with their immune systems. Stress can predispose a healthy individual to disease. Excuse me. Many of you know when you get stressed out or when you don't get enough sleep, that's when you're susceptible to getting a cold. Stress increases anti-inflammatory hormones and removes a natural barrier to infection. Diet can also affect host resistance. People who do not have the required nutrients are susceptible to infectious disease. One only has to watch TV or look to see where in the world infectious diseases are rampant, and there's a strong correlation in countries where people do not have enough to eat and higher levels of infectious disease. Also remember the dental caries at the beginning of the chapter. This is an example where diet allows infection to take place. So, we can talk about these natural barriers as the innate immunity. But we also need to talk about adaptive immunity, and then we can talk about some of the specific parts of it. So if I was going to talk about immunity as an overview to the whole idea of immunity, we really need to talk about the idea of self versus non-self, meaning the body's ability to tell its own self from things that are foreign to it. And this is one of the primary theories of immune theory, and is a very good way to sort of start thinking about the immune system. So one way to think about immunity is that your body is always checking to see that you are you, and that things that don't belong in you are not tolerated. As we said before, there are two types of immunity. There's the innate and the acquired, or adaptive. The word innate refers to something you're born with. An innate immunity refers to things that are always present and active on short notice. Innate immunity is called nonspecific immunity and can be conferred by the things that we just talked about. Physiological barriers like skin, mucous membranes, cough reflex. Chemical barriers like pH in your stomach, interferon proteins, enzymes like lysozyme and beta-lysin. And then 3, physiological changes like temperature and inflammation. Part of the innate immunity that acts on the cellular level are phagocytic white blood cells. And these white blood cells are important for trying to stop foreign material from growing or being in the body when it shouldn't. But they also are highlighted here in purple because phagocytic cells also do something very important, which is link the innate response to the so-called acquired or adaptive response. And without that specialized phagocytic cells that do that, we would never be able to stimulate the acquired or adaptive immunity, which allows us to maintain memory of being exposed to pathogens. So acquired immunity or adaptive immunity is obtained, and it's obtained when an individual is mounting a response to an invader. The cells involved with acquired immunity must come in contact with a foreign substance and then form a response that is specific to that substance only. So innate, we think of innate as nonspecific, and we think of the acquired as highly specific. The contact or immunization of a substance into the body triggers the activation of specific cells called lymphocytes in the acquired response, and then the subsequent production of proteins that react specifically with the foreign substance, and also allow the body to retain immunity to that substance in the future. All substances that elicit an acquired immune response are called antigens. Antigens activate specific cells called lymphocytes. Only cells with backbones or vertebrates have acquired immunity. And all substances that elicit an acquired immune response are called antigens, which I already said. So let's talk about the basic cells and the organs involved in the immune response. Both specific and nonspecific immunogenic responses are the result of cells that circulate through the blood and the lymph in the body. All these cells are derived from common stem cells. About 0.1% of blood cells are leukocytes, or white blood cells. These include the monocytes and the lymphocytes. In addition, cytokines released by these cells are molecules that influence the development of stem cells and determine which types of cells are being made. The whole blood is composed of plasma and these white blood cells as well as the red blood cells, and also proteins and other solutes that are involved with clotting, homeostasis, and the immune response. When you spin the blood down and remove the cells, you get the serum. So blood is primarily made up of non-nucleated erythrocytes or red blood cells. These are the cells that carry oxygen throughout the body from the lungs. In addition, as I said before, about 1% of blood cells are nucleated leukocytes or white blood cells. The white blood cells include many phagocytic cells that act both in the specific and the non-specific immune responses, as well as lymphocytes that mediate one of two specific adaptive or acquired responses. Blood also contains various proteins that act as clotting factors, hormones, antibodies, and signal molecules for the immune system. Myeloid, meaning bone marrow, precursors can develop into phagocytic leukocyte cells, including things like neutrophils, or also called polymorphonuclear cells, and mast cells, which participate both in the innate and acquired immunity, but have no antigen recognition properties. In other words, the purple here are all part of the innate immune response, And then a subset of these cells, the dendritic cells and macrophages, are going to link that innate response to this lymphoid cells over here that run the adaptive or acquired response. So these cells, these innate immune cells, are stimulated to act by contact with foreign material and the release of substance called cytokines by various cells in the body. Cytokines signal other molecules and further stimulate immune responses. As I said before, a subset of these leukocytes, such as the macrophages and the dendritic cells, may present after they're done swallowing stuff up, may present pieces of consumed material to T cells, which lead to an acquired or adaptive response. Two types of cells are derived from a common lymphoid precursor. So these are the lymphocytes. The two types of cells are T cells, named T cells because they mature in the thymus gland, and B cells, which mature in the bone marrow. T cells and B cells operate differently in the body, but both share the ability to react with very specific regions on foreign protein or antigens. So the immune system properties of recognition and reaction or specificity are held in the orange cells over here, in the lymphocytes. Nonspecific, innate, specific, acquired, or adaptive. So leukocytes are nucleated white blood cells, and many of them are phagocytic. Just to recap what we just said, myeloid cells are derived from a myeloid precursor, and they're divided into two categories. The so-called antigen-presenting cells engulf, process, and then present antigen to the lymphocytes, which links the innate and the acquired response. These cells are monocytes, macrophages, and dendritic cells. The other leukocytes, the other innate white blood cells, are called granulocytes. These contain toxins or enzymes that are released to kill target cells in extracellular killing, but also do phagocytosis. These include neutrophils, basophils, and eosinophils. These cells cannot present antigen to the adaptive immune cells. So, this first group participate in innate immunity and then link the innate immune response to the adaptive or acquired response, and these cells only participate in the innate response. All these cells are stimulated to act by contact with foreign material and or release of substances called cytokines by various cells. Cytokines are sort of like the hormones of the immune system. We don't call them hormones because they're not technically hormones, but they're signal molecules that further stimulate immune responses or suppress them. The other category we just talked about are the lymphocytes, which are specialized leukocytes involved exclusively in adaptive immune responses. And the two types are the B cells, which originate and mature in bone marrow, and the T cells that originate in bone marrow but mature in the thymus. The immune system properties of recognition and reaction, or so-called specificity, are held by the lymphocytes, the B and the T cells. Because they mature in these organs, both the bone marrow and the thymus are considered primary lymphoid organs. Now we can move on from the types of cells and talk about the lymphatic system. The lymphatic system is a separate circulatory system, and it drains lymph fluid from extravascular tissues, and sort of can be envisioned as kind of like the cleanup system or the cleanup crew in the body, right? So within this system, lymphatic organs are defined. Hold on. Sorry. Blood is pumped through the arteries and capillaries and returns from the body through the veins. In capillary beds, leukocytes and solutes pass from blood into the lymphatic system, and then that lymph nodes or nodules where the lymphatic system are connected contains high concentrations of lymphocytes and phagocytes, where we connect the innate and the adaptive immune responses. So lymphatic organs are defined as organs where the lymphocyte maturation, differentiation, and proliferation take place. So we're talking about the adaptive immune system. primary lymph organs where the B cells and T cells acquire antigen and recognizing ability, as we said before, are the thymus and the bone marrow. B and T cells, once they've matured, migrate from these primary lymphoid organs to peripheral lymphoid tissues such as lymph nodes, spleen, and tonsils. These secondary tissues is where antigen-driven reproduction or cloning and differentiation can take place if they're stimulated. So the thymus gland is where all these progenitor cells from bone marrow migrate and then differentiate into T cells. The thymus is a two-lobe structure that grows until puberty and atrophies thereafter. The thymus consists of epithelial cells organized in an outer or cortical structure and a central or medullary structure. The cortex has large numbers of lymphocytes of various sizes and some macrophages that are there to present or scavenge self-destructing thymocytes. T lymphocytes mature in the cortex and migrate to the medulla or the center where they mature further. The cortex contains these cells, like macrophages and dendritic cells, that facilitate something called thymic selection. And what happens during thymic selection is that these antigen-presenting cells, the macrophages and the dendritic cells, present self-peptides to all the maturing T cells. And if the maturing T cells react to proteins that are self, in other words, self-proteins, then they're induced to self-destruct. So most of the T cells that migrate to the thymus are told to commit suicide rather than being let out into the body where they can recognize you as a foreign invader. So this is one of the main areas where we sort of determine self from non-self. Everything that reacts to self in the thymus is told to just commit suicide, and the ones that don't react to proteins that are self proteins can then migrate out of the thymus into secondary tissues. So recognition occurs through something called the T cell receptor, which we'll talk about next week. And that T cell receptor is acquired during maturation of the T cell in the thymus. So we consider these primary lymphoid organs because it's the location for T cell differentiation and then selection, depending on whether it reacts to cell proteins or not. Most of this maturation occurs mainly during the fetal development of the organism, and completely mature T cells leave the thymus and then take residence in these secondary lymphoid tissues. And as I said before, only about 5 to 10% of maturing lymphocytes survive. So 90 to 95% are going to die in the thymus because they didn't tolerate self. Because they recognize self, we don't want them floating around the body. When that happens, it's called autoimmunity. The B cells do all their maturation in the bone marrow, right? And other vertebrates, such as birds, have specific organs for this, but ours happen in the bone marrow. In B cells, they differentiated from stem cells of the fetal liver, and later this function moves to the bone marrow, which is considered the primary lymphatic organ. Each mature B cell has antigenic specific receptors that have identical structure to the antibodies that the cell will produce after interacting with the antigen. In other words, the B cell receptor is a membrane-bound copy of the antibody that the B cell will make once it's stimulated after it leaves the primary lymphoid organ. Mature B cells, if they are not selected for destruction, are transported to the blood to secondary lymphoid organs where they react with antigen and then will produce antibodies if they do react. The secondary lymphoid tissues consist of structures that house these mature lymphocytes that have been stimulated by antigen to undergo further division and differentiation, such as lymph nodes, spleen, etc. So secondary lymphoid tissues consist of these structures that house the mature lymphocytes that are stimulated for further differentiation. Right? Secondary lymphoid tissues have two major functions. They trap and concentrate foreign particulate, basically the stuff that gets inside the body and then is destroyed, gets put into the lymph system, and then the lymph system is going to act as like the garbage chute of the body, and all this stuff is going to pass out through the body, and then on the way, that matter that has been destroyed by the body is going to be checked in these secondary tissues where the lymph connects to lymph nodes or spleen, right? And where it's checked for foreign material. And if that foreign material meets certain criteria, then you can stimulate a secondary or an adaptive immune response from all those lymphocytes that are hanging out there. So secondary tissues are the site of antibody production and also the site of T cell activation, with one major exception, which we'll talk about next week. And the major ones, as I said before, are spleen and lymph nodes, but they also include the tonsils, your appendix, and the clusters lining or sort of behind the mucosal tissue called Peyer's patches. The so-called mucosal-associated lymphoid tissue, or MALT, contains lymphocytes and phagocytes just like your lymph nodes, and it interacts with antigens and microorganisms from the gut, respiratory tract, and mucous membranes. The immune systems of recognition and specificity, as I said before, are held in lymphocytes in these tissues. And anything that gets into the phagocytized by the innate antigen-presenting cells and then presented there may or may not stimulate a further adaptive immune response. So this shows a picture of the circulation and extravasation or diapepesis between the blood circulatory system or pulmonary circulatory system and the lymph system. And mobile white blood cells can pass from blood to lymph or vice versa, either through these capillary beds where they squeeze out through extravasation, right, and into the other circulatory system, or by being pumped through the thoracic duct. So the two systems are intimate in this way and keep a check on both on each other. And this is where antibodies would be circulated into the bloodstream, and this is where materials could be leaving the bloodstream into the lymph, or vice versa, if they are stimulated to do so. So here's lymph nodes, a picture of a lymph node. They're comprised of medulla with many sinuses and cortex surrounded by connective tissue. Inside the lymph, germination centers contain mostly B cells that are undergoing mitosis or cloning. T cells are present in the deep cortical area along with dendritic or phage cells. The dendritic cells are going to present the foreign material to the T cells to activate them. And the lymph or lymph fluid, once the B cells are also activated, then leaves through the lymph node through the efferent duct. But the B cells never leave these secondary tissues. T cells, however, once they're stimulated, can change the proteins on the outside of the cell, if they're the right kind of T cell, and leave and migrate to the site of infection. B cells stay here and make antibodies, which are then circulated through the lymph and the bloodstream. This is a cell of a picture of extravasation or diapepesis. And here's the red blood cells in the blood system. Here are the lymph cells, and these are stimulated T cells that are passing from one side to the other. The way this happens is that once they're stimulated, the proteins on the outside of the cells change so that they can then stick to the endothelial cells lining the vasculature, and allows them to migrate from one side to the other. What causes them to move from one direction or the other is usually a chemical gradient of released by cells surrounding a wound site or an infection site that allows these types of cells to move from these secondary tissues, these T cells, into the bloodstream and go to the site of infection. So let's talk more about the innate, or more details about the innate cellular immunity or the non-specific immunity. And then we'll talk a little bit more about the specific or adaptive immune response before we talk about things that happen with the immune response. So we sort of talk about innate immune cells as non-specific, meaning that they don't react as specifically as the adaptive lymphocytes, right? So these are leukocytes, they're nucleated, they're circulating the blood, they're existing in certain tissues, and some of these tissues are part of the lymph system, and some of them just hang out in tissues underneath the skin or in your lungs in certain areas where they're adapted to hanging out in the specific areas. They're there no matter what you've been exposed to, so we call them innate. They're just part of the system that you always create, you always make these cells. And they sort of, one way to think about it is that they exist just beyond the boundaries, the physical or chemical boundaries of the innate barriers to infection. They have this ability to recognize and destroy foreign material and or its products. So they have this ability to do some recognition of what's foreign, what's not you. However, it's not as specific as the lymph cells, the T cells and the B cells. All right? So these cells don't require any previous exposure to a pathogen or its products. When a foreign object bumps into them, they have some proteins on the outside that can detect whether it's generally foreign, and that induces phagocytosis. Those types of things that are recognized are called PAMP, pathogen-associated molecular patterns, and they exist on the outside of bacteria. So examples of PAMP include things like LPS, or peptidoglycan, double-stranded RNA from viruses, or single-stranded RNA from viruses, and all of that stimulates these cells to swallow stuff up in a non-specific manner. So common phagocytes that circulate through the blood and the lymph include the neutrophils, also called polymorphonuclear leukocytes, or PMNs, because they have a fragmented nucleus. They're found in blood, bone, sites of infection. Other classes of phagocytes that do this are macrophages. Macrophages are large stationary phage cells that have the ability to present antigens to T cells and stimulate that specific immune response. So you find phagocytic cells in tissues. You find them in macrophages in tissues, especially lungs and underneath the skin. Excuse me. And also in lymph tissue. These cells together as a group are known as mononuclear phagocytic system, and so we have they used to be called reticuloendothelial system, but the name is not that important. But the macrophages can then differentiate into multiple types of cells that are location specific. So they're present in the lymph node and spleen, but they're also important antigen presenting cells. They're in your lungs, where they're called alveolar cells. And the ones that are circulating in the blood are called monocytes until they get to the mature stage. Once they're macrophages, they stick to tissue. So after swallowing up phagocyte, after swallowing up something foreign, all these phagocytes then have the ability to increase their affinity for that type of foreign material and become more efficient at destroying it. They do this by creating more of the proteins that stimulated phagocytosis to begin with. So this is what a phagocyte looks like in the textbook. It's this purple cell. It's got these lysosomes. It's got a nucleus that's not perfectly round. This is the picture of a macrophage cell. So what's a little bit confusing is we call these innate immune cells and we say they're non-specific, yet there is a recognition event that occurs for foreign material. So phagocytes have receptors on the outside of them that are involved primarily in the innate immune response that can recognize these broad structural motifs or patterns that are highly conserved within microbial species called pathogen-associated molecular patterns, or PAMP. PAMPs are generally absent from the host, so they're always recognized as foreign. Examples of PAMP, like I said before, including the bacterial cell wall and membrane components, peptidoglycan and lipopolysaccharides, certain sugars, and some nucleic acid motifs. Because they recognize particular global molecular patterns, These receptors on innate immune cells are called pattern recognition receptors, or PRRs. There's a number of PRRs that exist on phagocytes, such as macrophages, neutrophils, and dendritic cells. And then recent information has shown that cells involved in acquired immunity also contain PRRs. So to summarize, receptors on these phagocytic cells are involved in primarily innate immune response, recognize these broad motifs called PAMPs. And PAMP is things that are recognized as foreign, but not specific. So in other words, it might, PAMP is the LPS on gram-negative bacteria, but not specifically on E. coli versus something like Salmonella, right? Or PAMP is the peptidoglycan on the outside of a gram-positive cell. And those PAMP is recognized by pattern recognition receptors. We'll come back to that in a little while. Now we can move on to the overview of adaptive immunity. Adaptive immunity is the acquired ability to recognize and destroy a particular pathogen or its products. So now we're recognizing things down to the molecular level. Unlike innate immunity, it's dependent on previous exposure to the pathogen or its products. So there has to be a recognition event where the adaptive response is stimulated to respond to a specific pathogen. And then once that recognition event occurs, which can take days, further exposure will result in the adaptive immune response. So this is very specific. It's directed at an individual molecular component of the pathogen, or a so-called antigen on the pathogen. So once antigen is presented, the primary immune response can occur, which is that recognition of this foreign material by the adaptive immune response. That results in stimulation of specialized antigen-reactive leukocytes called lymphocytes, the T and the B cells, which we talked about previously. Each lymphocyte produces a unique protein that interacts with that single antigen. On T cells, the ability to recognize this foreign material is in the T cell receptor with various other proteins, but the primary one is called the T cell receptor. And the response is said to be cellular because T cells then release chemicals that act on other cells. In B cells, there's a B cell receptor. Once stimulated, and the B cell is a signal is confirmed through various modalities, those B cells then change to make soluble versions of the receptors called antibodies, or technically called immunoglobulins. And we call that antibody-mediated, or historically the humoral immune response. So acquired immunity can be broken down into these two major branches: antibody immune response by B cells that release antibodies, and cytokine-releasing T cells called cellular. After any of these cells are stimulated, the lymphocytes clone themselves and amplify a specific immune response directed at cells that have shown that particular antigen. So once stimulated, it's a very powerful force to remove a particular pathogen from the body. In fact, it's so strong that if you prime the system and you get a secondary exposure, it can be strong enough to prevent disease from happening, which is what we try and do when we immunize people, either through getting the disease, like infecting your kids with chickenpox, or by giving them a vaccine for something like chickenpox. So we're going to have these interactions with immune T cells with this foreign material or antigen well, it says uninfected cell. The antigen's not on. The antigen's infected cells, or cells that are causing infection, or antigen that's being presented on these specialized phagocytic cells called antigen-presenting cells. T cells then recognize the antigen really only when presented in self-proteins called major histocompatibility complex, or MHC proteins. So it's not enough that the foreign material is presented to a T cell. It has to be presented in a very special molecule, and depending on which form of this MHC protein it's presented in, you'll get different types of T cell responses, because different T cells respond to different foreign material or antigen that's been presented in different major histocompatibility complex. So this is what it looks like in a very simplified version. And we'll go into some more detail next week in class, but just to give you an overview. So here's your it looks like a macrophage. Here's your antigen presenting cell. It's going to get stimulated to swallow up the foreign material, in this case bacteria or viruses, and it will kill them. It will destroy that material. And then, because it's an antigen presenting phagocyte, an APC, it will then present pieces of proteins that are comprised of this material on the outside of the cell, and these proteins called MHC. And there's two types, MHC class 1 and MHC class 2. And depending on which type the protein is presented in, you'll stimulate different types of T cells. Okay. So we have this antigen, the antigens, the purple one here and the blue one here, right? Corresponding to purple and blue of these bacteria, right? And it's going to get, these cells are going to be destroyed and cut up, and then pieces of foreign protein get put into these major histocompatibility complexes. And T cells can only recognize foreign protein when it's in one of these two proteins on the outside of the cell. If and only if a T cell receptor recognizes antigen presented in this protein called MHC, then the cell is stimulated to release chemicals. And the chemicals released have to do with the type of T cell stimulated. And the type of the T cell stimulated has to do with which type of MHC the antigen's been presented in. So this is sort of the crux of linking your innate immune system to an adaptive or specific response. And the whole thing is dependent upon whether this T cell receptor can stick or bind to this protein with this antigen in it, presented in this particular molecule. So for example, there's two types of MHC, and we'll go into more detail with the MHC next week. But class 1 MHC is on all of your cells, all your somatic cells. All of your nucleated cells have class 1 MHC. And this type of MHC presents proteins, foreign proteins that were generated inside the cell, so an intercellular parasite. And this is one of the ways that we sort of tell our immune system that we're us. So when it doesn't have foreign material in it, it's always presenting self-peptides. And the self-peptides are not recognized by your T cells as foreign, so it's a way of your T cells sort of just bumping into you and going, oh, you're fine, you're okay. When one of these peptides gets put in here that's foreign, a type of circulating T cell called a cytotoxic T cell may recognize it. And when I say recognize, I mean it may fit in here like a key in a lock and hold that T cell next to that antigen presenting cell for a period of time. And what that does is then induce a signal to be transduced inside the T cell, and that cytotoxic T cell will then release all these granular enzymes that then destroy the cell that presented the antigen. So antigen presented in class 1 MHC is a way for the sick cell to tell your immune system that I'm sick, kill me. Right? These T cells, these cytotoxic T cells, they do not exist in the lymph and secondary tissue. They are circulating constantly, always bumping into your class 1 MHC saying, are you you? Are you okay? Are you you? And when that signal says no, I am foreign, and there's recognition, they release proteins that will then kill the cell that is presenting the antigen. The second type of antigen recognition event is the type of antigen recognition that we talked about from the antigen presenting cells. And unlike MHC class 1, only antigen presenting cells have class 2 MHC, meaning only things that are phagocytic, that swallow up foreign material from outside the cell, kill it, and then present it, have class 2 MHC. So these include the macrophages, the dendritic cells, and as I'll show you later on next week, the B cells as well. And so this is a specialized MHC that only exists on immune cells in the body. And when the foreign when the phagocytic cell that's antigen presenting bumps into that foreign material and swallows it up and kills it, it will then take that foreign protein that's made outside the cell, exogenously outside the cell, and present it in class 2 MHC to a type of T cell called a helper T cell. And depending on where this happens or what antigen presenting cell this occurs to, one of two events can occur. So if a macrophage presents it in class 2 MHC, this foreign protein, then the type of helper T cell that interacts with it is called type 1 helper T cell. And type 1 helper T cells only recognize this foreign material when it's presented in class 2 MHC on a antigen presenting cell like a macrophage. That stimulates them to release a set of cytokines that's specific to type 1 helper T cells that induce or increase inflammation. So in this case, those chemicals that are released will act back on this macrophage cell, cause it to clone itself and increase its ability to go out and look for that same pathogen. At the same time, it will stimulate vascular dilation, inflammation, sometimes fever, depending on the complete makeup or how strongly it's stimulated. But the important thing here is that these cytokines that are released also act back on the cell that's stimulating them. So it's a local event. It's a very strong event. They will act back on here. The other thing that happens is that in both these cases, the stimulation of these cells causes them to clone themselves and go out and look for those clone cells go out and look for other macrophages that are presenting the same antigen. So the whole system is amplified. What's not shown here is B cell stimulation, which has to do with another type of helper T cell called type 2 T cells. So just so we go back here just to be sure, this antigen recognition reaction is the key to stimulating your adaptive immune response or acquired immune response. Without this event, and this can take days, either in the lymph nodes or in specific tissues, you're not going to get the adaptive system responding. It's a very sort of specific key gateway because you don't want this part of your immune system to go off incorrectly. You want to be able to tolerate what you need to tolerate and respond to foreign material that's damaging the body. And if it went off all the time, your inflammatory response would be completely out of control and you would have serious autoimmune problems. So the antigen presenting and recognition event is the key to all of this. So we have these two subsets for now of lymphocytic T cells. There's the cytotoxic T cell, which recognize antigen presented in class 1 MHC only on an infected cell. So if your cell gets sick by an internal parasite like a virus or some specific bacteria that can do that, that foreign material that's generated inside the cell, endogenously inside the cell, can be presented in class 1 MHC by that cell, and then these T cells will recognize it and release chemicals that will kill the sick cell. The other general category we've talked about so far are these helper T cells. They only interact with antigen or peptide in class 2 MHC complexes, and these types of MHC molecules are only on antigen presenting cells, so specialized white blood cells in your body. These are on all of the class 1 MHC are on all of your nucleated cells. These are only on immune cells. These cells get stimulated and act by releasing cytokines that promote different immune reactions depending on which helper T cell they are and which antigen-presenting cell stimulated them. Within the T cell lymphocyte, we have two subsets we talked about before. The diagram that showed the helper 1 T cells, which can initiate inflammation and immunity by activating more macrophages. And then this other subset called the helper 2 T cells, which are involved with B cells that produce antibodies. So this is I think it's a tubercular test. I'm not sure, but this is a picture of inflammation mediated by stimulation of antigen-specific type 1 helper T cells and macrophages during a tuberculin test. The strong reaction indicates previous exposure to tuberculin protein antigens because you won't get that type of inflammatory response until there's been a previous antigen recognition event. So I'll show you this on a graph, but there has to be this whole primary antigen recognition event and response before you would get this quick, large immune response of inflammation the second time that it's stimulated. So let's talk about this other branch of immunity, the B cell activation. Immunoglobulins, as I said before, are soluble proteins made by B cells once they've been stimulated through an antigen recognition event. B cells display antibodies on their cell surfaces called receptors that can directly interact with antigens that then cause the B cells to ingest the pathogen through phagocytosis. In this case, the B cell is going to act as its own antigen-presenting cell to the T cell. Right? So, this should say present. B cells can then present many pathogen-derived peptide antigens that are presented to antigen-specific T cells. So how does this look? So here's a B cell, and the B cell would be in a secondary tissue like a spleen or a lymph node or a Peyer's patch, malt tissue, something like that. And it's got a B cell receptor on the outside, right? And this B cell receptor can bind to foreign material that's been passed through the lymph. It could be destroyed bacteria, could be live bacteria. And if there's recognition, and only if there's recognition between the foreign material and this receptor, it will then be induced to take the whole protein complex, including the foreign material, inside the cell, where the foreign material is broken down and then presented in class 2 MHC. So the B cell is its own antigen-presenting cell, right? And it's class 2 MHC because it's taking stuff from outside and presenting it versus stuff that was grown on the inside. Now, in the lymph node or spleen or secondary lymph tissue, the only cell that can recognize foreign material presented in class 2 MHC on a B cell is this subtype of helper T cell called a type 2 helper T cell. Type 2 helper T cells are hanging out in these nodes, right? Constantly bumping into these cells. And when they bump into something and they stick, it's called recognition. And if it sticks, it means that the T cell receptor also recognizes this piece of antigen or protein that's been presented by the B cell. And a signal gets transduced into the cell, and then a different subset of cytokines are released to then act upon the antigen-presenting cell. And in this case, we're not stimulating a macrophage to clone itself and induce more inflammation. We're stimulating a B cell to then transform itself, clone itself into so-called plasma cells that make soluble versions of this B cell receptor called antibodies. These antibodies get released out into the lymph and then the blood, and wherever they run into this foreign material, they stick to it. So they act like flags or identifiers to the rest of the immune system saying, this stuff is foreign, get rid of it. And that's really the function of antibodies in a very simplistic way inside the body, right? So all this happens inside a secondary lymph tissue, right? None of it happens outside. They never leave the lymph tissue. The only thing that leaves are the antibodies that are produced. But again, you have to have this recognition event. This event could take 5 to 7 days. You could have a full-blown inflammatory response and innate immune response going on. And depending on whether you've been exposed to the pathogen or not, you need some time to develop this primary response. Once the cell is set and is making the antibodies, it's not shown here, but a subset of these will be kept as memory cells, which don't have to go through this whole antigen recognition event again. So let's talk about what antibodies are, right? Antibodies are proteins that are made by B cells that have been stimulated by class 2 helper T cells through this antigen recognition event. They then differentiate into plasma cells that produce soluble versions of their receptors called immunoglobulins, and that's the primary antibody response. After infection, if you subsequently expose that same organism to the same antigen, some of those cells that were made during the primary response act as memory cells, and they hang out in the lymph. And if you get a secondary exposure to that same pathogen, they no longer have to go through that whole antigen-presenting event with the T cell. Instead, they go directly to the plasma cell event and start dumping antibody into the lymph and the blood. So this is sort of what it looks like in the B cell response. Here's the primary response. Here's the secondary response. And what we're looking at is antibody in the blood or lymph. So you have an event where a B cell is stimulated to present antigen to a T cell, and that B cell goes through recognition phase and starts making antibodies, and the body is cleared from infection, right? Or you could say this event might even be through an immunization, through an artificial immunization. Anyway, the primary response occurs. It takes a certain amount of time, and over that first couple of weeks, you see an increase in antibodies in the cell. That level of antibody stays in the cell. Over time, it slowly goes away, right? And then if that organism is challenged again with the same pathogen, with the same antigen on it, there is no need for this primary response to occur again. Instead, a subset of cells that were previously stimulated directly clone themselves into a large plasma cell event that dumps antibody into the blood and the lymph, which is the secondary response. This is the response you're trying to stimulate when you're trying to prevent infection by pre-exposure. So by dumping all this antibody into the bloodstream, you can prevent infection because it's such a strong and swift response to something that the body has previously seen. So the property of memory is specifically within the lymphocytes. We have specificity, now we also have memory. And there's also memory with T cell responses as well. Once the B cells are stimulated, they can make an antibody that specifically sticks to that protein they were stimulated to bind with anywhere in the body. And it's very, very specific, and it actually improves its specificity over time as they get stimulated over and over again for a specific pathogen. In addition, each of these antibodies that are made can have a different class that's associated with it. So the specificity is always going to be the same for the protein that was presented to them, so the part that binds to the foreign protein. But the other end of the antibody can be made in different ways that are called classes. And each class determines where the antibody works in the body, and partially determines also what immune cells that antibody stimulates once it's stuck to foreign material in the body. So, for example, immunoglobulin mu and gamma are IgM and IgG are circulating antibodies in the blood and the lymph. IgA is a secreted antibody on the outside of your mucous membranes. IgE is circulated, but is involved in parasite immunity and also in hypersensitivities, allergies. And IgD is not circulated. It is only found on the surface of B cells, so it is not a circulating form of the antibody. So, as I said before, once an antibody sticks to a foreign protein that it recognizes in the blood or the lymph, it acts as a target for other parts of your immune system to come and destroy that foreign material. And part of that targeting proteins are called the complement system, which is part of your innate immune system, that only binds to cells that are flagged by antibodies or by other proteins in the cell. Once the complement system is flagged, or other phagocytic cells are flagged, they bind to the cell and they destroy them, either through lysis or opsonization, which is basically just making the cell recognizable to phagocytic cells. So we've got antibodies, they run around, they bind to foreign material. They can also bind to toxins. They can inactivate toxins or neutralize them. Here's a picture with a lot of activity in it that we haven't talked about yet. But in general, what you're seeing here are antibodies that are binding to their foreign material on a bacterial cell. The FCR region is the region that determines their class. The red region is the variable region, which determines which protein they're going to stick to, which foreign protein they're going to stick to. Once they bind here, here's a phagocytic cell. There are other proteins that can then recognize this cell as foreign and induce phagocytosis. So that's one thing that's going on. The other thing that's going on is that the binding of these antibodies to the cell induces a bunch of proteins called complement to drill holes in the cell. Complement acts as a handle for phagocytosis as well, but can also destroy some gram-negative cells. If enough holes have been drilled into it, the cell will lyse and be destroyed. And then finally, some of these complement proteins act as binders for other cell receptors on phagocytes that also induce phagocytosis, as well as inflammation. In addition to identifying foreign material for phagocytosis or the complement system, antibodies can neutralize probes, can bind to toxins in the cell that would normally cause cell damage. So, for example, when you get a tetanus shot, you're not getting a shot to stimulate your immune system against the tetanus bacteria. You're getting a shot to stimulate your B cells to make antibodies against the tetanus toxin. And when these antibodies are circulating in the blood, right, they maintain themselves for a number of years. If you're exposed to that tetanus toxin, they bind to it before it can damage you and protect you from the toxin of the tetanus organism. That's called neutralization. So let's talk about, that's sort of a general overview of how the innate and the adaptive immune responses occur in the body. Now we can talk about some generalized host defenses and also some hypersensitivities. So inflammation is a non-specific reaction to stimulus that's foreign or harmful. It can be mechanical or it can be infectious. Inflammation occurs when phagocytes are stimulated to swallow stuff up. They release cytokines. And then also when class 1 helper T cells are stimulated to release cytokines that act on other cells. Inflammation is stimulated when complement is stimulated by antibodies. So in general, it's a non-specific reaction. It causes redness, swelling, pain, or edema, and heat localized at the site of infection. And the mediators, as I said before, are these proteins called cytokines and a subclass of cytokines called chemokines, which attract other white blood cells to the site of injury or infection. Effective inflammatory response isolates and limits tissue damage, destroying damaged cells and pathogens. And inflammation, when it works correctly, is primarily responsible for isolating things and making sure they don't circulate to your body and form new sites of infection. But as we know, if it's out of control, it could also damage healthy tissue, or if it goes on for too long. So turning the system on is just as important as turning the system off. So here's two induced types of inflammatory reactions. This is a reaction to a smallpox vaccine, Vaccina with a vaccinovirus. That's why vaccines are called vaccines, because vaccina is cowpox. That's vacca is cow in Greek and Latin. And we stimulate the immune responses to cowpox, which also protects us from smallpox, or we did. Many of you are too young to have done this. And you get this huge inflammatory response at the site of injection, where you can. Over here on the right is a Actinomyces infection that's in the center here, the bacteria growing, and all the neutrophils that have been called to the site, of infection and migrated to the site of infection by the release of cytokines from white blood cells. So they release, they increase their phagocytosis of the site of infection. There's sort of a battle going on here to see who will win, but all these white blood cells then migrate to join in and try and get rid of the inflammatory cause of inflammatory stimulation. So in general, what happens is the neutrophils are the circulating neutrophils, so they're not antigen presenting, are the first arrived by infection. They get attracted to cytokines called interleukins, which are released by the endothelial cells and the epithelial cells that have been damaged. And most of these cells involved in the inflammatory response are phagocytic, right? But and so are these, right? These so-called neutrophils or polymorphonuclear leukocytes in the first half hour to an hour. If the cause for inflammation persists beyond this point, the site becomes infiltrated by mononuclear cells, including macrophages and some lymphocytes, within about 56 hours. Macrophages help the neutrophils clear the source of inflammation, but also help initiate the acquired immune response by presenting antigen to lymphocytes, which then ramp up inflammation. If injury or invasion is caused by microorganisms and continues, the inflammatory response will be supplemented by acquired immunity, antibodies, cell-mediated inflammatory responses, etc. The acquired immune responses trigger specific processes and also further stimulate parts of the innate immune system. So there's release of cytokines by helper T cells stimulates the non-specific white blood cells to increase phagocytosis. So we have activated neutrophils. They release chemokines, recruit macrophages, guiding them along a gradient, chemical gradient that they're releasing, so chemokine gradient. And then the usual response is rapid localization and destruction of the pathogen. When fever accompanies inflammation, it's caused by the release of pyrogenic compounds such as interleukin-6 and interferon and tumor necrosis factor alpha. All of these are cytokines, and they get released from monocytes and macrophages in response to certain types of compounds, like endotoxins of gram-negative bacteria. In some cases, inflammation fails to localize the pathogen, and the reaction becomes widespread. This can lead to septic shock, life-threatening conditions. Not good. Right? So if you have a systemic inflammatory reaction, you can get serious consequences: uncontrollable fever, uncontrollable inflammation, drop in blood pressure, and still to this day, death of 30% of individuals that go into septic shock, though we're much better at controlling it that we're doing. So it really isn't the infection that might kill you in this case. It's the overlarge response of your inflammatory response that can put you into shock and cause problems. Let's see if I can get this to play. Yeah. Let's see. Bear with me. I don't want that. Well, I'm gonna have to show this to you in class. Sorry, I'll show it to you next week, or if you remind me, I'll show it to you in lab. You should be able to see it on yours. I can't show it while I'm recording for some reason. So that's inflammation. Now we can talk a little bit about immunity and immunization. Animals normally develop two types of immunity: natural active immunity, where you get an infection that initiates an adaptive immune response, and then you develop a secondary response, and that's strong enough to prevent infection when you're exposed again to that pathogen, and that's actively gained. The other way animals, including us, can get immunity naturally is through passive immunity, passed through the mother to the fetus, through the placenta, or even a little bit in breast milk after the baby's born. In this case, immunity is given to the fetus or the neonate that from the mother's memory, or the mother's antibody system, primarily antibody system, although you learn more about this every year, where more and more types of things can cross over the placental barrier that protect the baby. Other ways of getting immunity are artificial, and artificial immunity can be gained actively through vaccination. So in this case, you stimulate your antigen-presenting event, or what we're trying to do is stimulate your antigen-presenting event when you get a vaccine, say for influenza, where phagocytic cells swallow up the foreign virus particles and present them to T cells. In the case of a virus, you're trying to use attenuated live virus is the best kind, virus that doesn't make you sick, but it infects the cell anyway. And then the type of T cell response you're trying to stimulate is the cytotoxic T cell, because those are the types of T cell responses that can prevent you from getting sick when you're exposed to influenza next season, or that season even. So you get a shot of live attenuated influenza vaccine. It infects some cells in your shoulder. Those cells start presenting antigen and class 1 MHC. Instead of self-antigen, they put in some virus proteins, often from hemagglutinin and neuraminidase. And those proteins get recognized by cytotoxic T cells, which then release things that kill them, but also they clone themselves, and some of those cells remain as memory cells. And then when you're exposed to that same antigen again on a live virus from, let's say, a pathogenic strain of influenza, the T cell response is so strong it doesn't have to go through all of that antigen recognition again. When it sees the virus, it clones itself and ramps up this huge immune response, preventing disease from occurring, even though you've been infected. And that's active artificial immunity. You can also do this passively. They don't do it so much anymore, but let's say you lived in the Southwest or in Australia where spider bites and snake bites were a big problem. Immunity from these things is usually not acquired so well because sometimes they're fatal. So, these types of animals are harvested for their venom, and then that venom is diluted and given to laboratory animals who develop an immune response to it. And then those antibodies from those animals are collected and frozen into sera, so that if you get a bite from a snake, you get injected with the serum that has antibodies against that particular toxin, and you can neutralize the toxin so your body can get rid of it before it causes too much damage. They used to do this type of thing for certain diseases before antibiotics too. You can types of pneumonia caused by pneumococcal disease were prevented through antiserum. I'm getting this to sorry about the phone. Someone will pick it up. Okay.
Is that just driving home from clients, worried about the election, and I hope you're well.
Sorry about that. It's my sister-in-law on the phone. We're back. So this whole idea of immunization is where we're generating the artificial active immune response by exposure to antigen or antigen mixtures in the form of vaccine. And if you use live virus or live cells, you usually get a more effective response because you get more effective antigen presentation, especially for viruses where you need to stimulate your somatic cells to present antigen to cytotoxic T cells. However, whenever you stimulate through vaccine, you're stimulating not just one part of your immune system, but multiple parts. So even if it's a virus, you stimulate the antibody response. And even if it's a bacteria, you can stimulate some inflammation as well as antibody response. So I don't really get to talk. I don't really test you on the importance of immunizations, but it's kind of an interesting subject, especially because of the amount of information out on the internet, good and bad. But there is a lot of data out there about immunization, and I really strongly suggest that you do some research and vet your sources of information very clearly so that you know what is in fact data-driven versus what is anecdotal, because there's a lot of misinformation about immunizations out there as well. So we can establish the importance of this. Most of us don't remember prior to the idea of immunization, prior to when people got polio and people got mumps and babies died of measles and huge birth defects and things like that. So we have a sort of a collective memory that is missing, but clearly my parents' generation grew up with these things, and so for them, vaccination was a bit of a godsend compared to the amount of human suffering that was going on. And now we have a different discussion about it. So it's a lot about risk perception in the population. Sometimes you only need to be vaccinated once. Some things that you get for life, like chickenpox. And so you're constantly stimulated against it. And then some things you need a booster shot for. And the difference there usually has to do with whether or not you're using live virus or live bacteria, or whether or not that organism is like influenza, which changes every year. So the proteins change every year a little bit. So your immune system gets fooled because the pathogenic or virulence factors on these organisms are always trying to gain an edge, and as you are always trying to protect yourself. So it's always in flux. So when you get a booster, it's more often because the thing that you're being immunized against is not generating a strong immune response, or you're only generating an immune response in part of your immune system, say the antibody response versus the full T cell response. This is a picture of recommended vaccines. I don't know what to say about this except everybody should get their polio vaccine. And if you haven't, then they've actually added this on here. I'm too old to get one of these. It's all over for me, but anyway. And they even have rotavirus in here. I mean, a lot of these things, it's all optional except when you come into school, when you need measles, mumps, and rubella, or you can't participate in certain public activities because you could be a carrier. All right. Anyway, I'm not going to test you on that. I just left it in there because they brought it up. It's an interesting topic. Besides just using attenuated vaccine or dead viruses or dead vaccine, dead things, there are different approaches. A lot of this research is being done because of certain viruses like HIV, and new immunogens are being developed through all sorts of genetic engineering. Synthetic peptides have been used successfully in cases where the entire structure of a pathogen or protein is known, so you don't have to introduce a pathogen into the body. Genes from viruses get incorporated into less pathogenic viruses, and then people are immunized. Genes from pathogenic proteins can be introduced into yeast cells that will then make the pathogen peptide. That's how Hep B vaccine is made. And also, DNA vaccines are being developed. And these vaccines take genes from a pathogen, incorporate them into a plasmid, and then the plasmid is introduced into the host where it's transcribed and translated into protein. These proteins then get presented to the immune response, and what's nice about DNA vaccines when they work is they stimulate both the CD8 and the helper T cell and the cytotoxic T cells, and they're the only synthetic vaccine that can stimulate B cells and all the T cell response, and therefore you can get a stronger, longer lasting response. So sometimes the immune response goes wrong and can produce negative or even fatal results. When we talk about hypersensitivity reactions or allergic reactions, we're usually talking about things that are stimulated by antigens that we call allergens. So the antigens are foreign material that the body should tolerate but doesn't. It reacts immunologically to. And sometimes those proteins are from outside the body, and sometimes, very sadly, they're proteins from inside the body. And we'll also talk about the last class of exotoxins, which are superantigens that also overstimulate your immune response. So hypersensitivity reactions are defined as those that are exaggerated or damaging to the host, but the cellular and molecular mechanisms are the same as immune responses that protect the host. Hypersensitivity is that inappropriate immune response that results in host damage, right? And they get categorized according to the type of material that stimulates it and the mechanism that produces the disease. So in general, they're classified as in 4 types of hypersensitivity. Type 1, also called immediate, is stimulated through the release of IgE antibodies, and it's what we normally consider allergy, right? And type 2 is cytotoxic, is stimulated by the production of IgG antibodies that interact with self-proteins on cells, and this can cause cytotoxic effects in the body where the immune system destroys your own cells. Type 3 is the failure to clear immune complexes from the body, where IgG antibodies form clusters regularly with complement proteins, and then that material is supposed to be removed from the body through phagocytosis. When that doesn't occur, it can form these inflammatory stimulating particles that circulate, get stuck in organs like the kidneys and cause tissue damage. And type 4, which is called delayed, is an inflammatory response, and instead of antibody, it's mediated through type 1 helper T cells. And some immune diseases are clearly one of these types, and some are combinations of these types of responses. So IgE-mediated reactions are stimulated by the binding of IgE to immune cells called mast cells, which are stationary cells in tissues, and right underneath your epithelial lining. This can be a quick reaction, but usually there's some priming of the system first, so you have to have an antigen recognition event, and they can be annoying, like hay fever, or they can be life-threatening, like food allergies. And about 20% of the population suffers from this hypersensitivity in their IgE production of antibody. So this is what it looks like. You've got a B cell that's binding antigen that it should normally tolerate. It's presenting that antigen, and the helper T cell is recognizing and releasing a set of cytokines that stimulate it to make a plasma cell. In here, there are chemical conditions that make it form IgE antibodies instead of IgA or IgG antibodies. And IgE antibodies circulate, but they then stick to these receptors in mast cells. And this does two things. One, it prevents them from performing like IgG, like identifying foreign material the same way to complement, if you would. But it also extends the half-life of these. So once you have an antigen recognition event, these things are made. They might be broken down within a week or two, but once they're stuck here, they prime these cells for a much longer time. So it sensitizes these mast cells by binding to them. Then when the mast cell is exposed to that foreign particle again, the two antibodies can be cross-bridged, and that induces a signal to be transduced across the membrane, which releases all of these granules with bioactive materials like histamines, serotonin, leukotrienes, and all of the stuff that makes your eyes itch, your nose water, and induces inflammation. And if it's a mild reaction, you can have hay fever and asthma, and if it's a huge reaction, you can go into shock or get closage of air passageways, depending on where these things are. So we say these are allergy events, and this is how they work. So there's a lot of theories about why this happens, but most people, if you look at see where the normal function is of IgE antibody, you'll see that it relates to worm parasites. So these are the types of antibodies that are you get in response to worms, and we don't really stimulate our immune system that much with parasites like this anymore. The water we're drinking is primarily safer, so this is sort of a misdirected part of our immune system that is looking for a home, I guess, is one way to think about it, in the people that have this reaction. So things that stimulate this are pollen, insect venom, food, dander, dust mites. People who have this know, as I do, what it is. Type 2 is cytolytic or cytotoxic hypersensitivity. These reactions occur when circulating blood antibodies, IgG or IgM, bind to antigen on cell surfaces and initiate a complement cascade that causes cell lysis to self cells, right? Examples include myasthenia gravis, Goodpasture syndrome, and also involved in juvenile diabetes. Type 3 hypersensitivity occurs again when IgG or IgM circulating antibodies complex antigen and then activate complement cascade in tissue or in circulation, and the complexes are failed. You can't get rid of them, so they attract white blood cells that release lytic enzymes and damage cells, and this can happen within hours. And the poster child for a type 3 reaction is lupus erythematosus. Where am I? Systemic lupus erythematosus, right? Type 3. So here are some diseases with their types. These are all type 2, type 3. Pernicious anemia also. So examples of type 3, autoantibodies that cause Hashimoto's disease, juvenile diabetes. There's also, where is diabetes? Can't find it. It's 2 and 4, so you see it's complicated. Another complicated one is rheumatoid arthritis. It says it's 3, but all sorts of things are going on with rheumatoid arthritis, where complexes form in joints and then stimulate all sorts of inflammatory responses. So type 2 and 3 hypersensitivity can lead to immune reactions against self, and these autoimmune responses can cause serious disease. The type let's see. So this is what I just sort of said. In some cases, you can make antibodies against self, as in with lupus. Sometimes you get anti-DNA, and that attacks us. And then the fourth type is the inflammatory type, which we call, fourth type is delayed hypersensitivity, and this has to do with inflammatory helper T cells. It's called cell-mediated. It's characterized by tissue damage due to inflammatory responses, and symptoms appear hours after secondary exposure, so you also have an antigen-presenting event. And typical antigens are microbes, some self-antigens, so autoimmunity, chemicals that bind to skin that create new antigens. So things like poison ivy is a type 4 hypersensitivity, because the compound that's released from poison ivy is not immunogenic in itself until it attaches to your cells, changing the proteins on the membranes, which then develop an inflammatory immune response from being foreign once they've been modified. Some drugs act this way too. All right. Finally, in this chapter, we'll touch upon superantigens, and we talked about exotoxins the last part of the course, and exotoxins are important. We talked about the cytolytic toxins and the AB toxins, and then the third category are the superantigens. So these are proteins that are released by growing bacteria that act as virulence factors in the body. And what they do is they stimulate your specific or adaptive immune response in a non-specific way. So, for example, when you turn on your immune response, your adaptive immune response, something like 0.0001% of your T cells might be stimulated to clone themselves and help you get rid of a pathogen or prevent infection in a secondary response. When the superantigens are released inside the cell, they come up to the T cell receptor and the MHC, and they bind the outside in a non-specific manner. So instead of turning on 0.0001% of these T cells, now what you're doing is holding the T cell receptor and the MHC of adjacent cells together after they bump into each other without the proper antigen stimulation. And when this happens, the T cells get turned on, and you have massive inflammation due to what's called a cytokine storm. So instead of turning on a small percentage, you turn on 15-20% of your adaptive immune response, and you can go into shock quite easily. And different bacteria have these types of things. Strawberry tongue is from superantigens from strep. Toxic shock syndrome from staphylococcus causes this. A bunch of other ones can do this. So that's where I'm going to leave you
now. And then next week, we're going to go over some of these same concepts again, with a little bit more detail, focused on antigen presenting, phagocytosis, antigen presenting, and stimulation of T cells in the adaptive response.